So, ladies and gentlemen, here we are in the heart of London at Shakespeare's Globe Theatre. And why are we here? Well, because I wanted to take you here and see this magical place. But it is utterly irrelevant to what we're doing today. So follow me, please. How would you describe your field of research?
Well, what I would say is a simple question: how does the brain store information? How are memories stored in the brain?
Right.
Given that we know the brain consists of a huge number of nerve cells and the connections between them, what happens to those connections, all those cells, when you lay down a memory? Because something must happen. The brain must change in some way because it now has this memory which it didn't have before. So, tomorrow I will look back on today and remember this interview with you. My brain has changed in some way. There has to be a physical change. So the question is, what is that physical change?
Right.
And long-term potentiation is that physical change. It's a change in the efficiency of the connections between cells in a subset of cells which are storing this memory.
When I got started in research, I began by working on learning and memory. But at that time, all we really had was a range of tasks that you could do with people or with animals, and to try to investigate what brain areas might be involved. But we were quite unable to have any sense of how a particular brain area might do something. We could say the hippocampus is involved, but we didn't know how.
It was obvious that the hippocampus was the place to look for two reasons. One, it's simple, and secondly, we knew it had something to do with memory.
I trained as a pharmacologist in the University of Bristol. Right. And there was a major interest in glutamate receptors. So I was aware of the emerging concept of different types of receptors which receive the signal from glutamatergic neurons. Right. And when I went to become a postdoc scientist in Vancouver, Canada, I was there demonstrated to me the process of long-term potentiation, the phenomenon that Tim Bliss had discovered about 10 years earlier. And I was just fascinated. I saw this process for the first time and said, this is what I want to work on. This is just incredible. As a physiologist, it was the most striking thing I'd ever seen.
I worked on a big book on the hippocampus together. So we've known each other, of course, for a very long time. And it was in the early 1980s, some 10 years after the original publication of Bliss and Lermo describing the physiological effect, that Graham discovered the importance of a particular type of glutamate receptor.
And I should say there's also, depending on the pattern of activity, you can actually get a decrease in synaptic efficiency, which is the opposite of long-term potentiation, known as long-term depression. And that's also activated by the NMDA receptor, but that engages different proteins. And why we're interested in that is, firstly, it's very relevant for learning and memory, which is, of course, what Richard Morris has shown. Yeah. But it's also very relevant for cognitive deficits, which occur in disorders such as Alzheimer's disease, autism, schizophrenia. In fact, any brain disorder which has a cognitive deficit component is likely to involve a dysregulation of the process of long-term potentiation.
And then Richard used that finding and that particular antagonist that Graham had used to block the NMDA receptor to show that if you block the NMDA receptor in the learning animal, then it learns much more slowly and cannot remember what it's learned.
Right. I thought, my gosh, this is terrific. We can perhaps actually go beyond just asking whether a particular brain area is involved to trying to ask how it was involved. So for me, it was like, a real turning point. It's a really very exciting time for young people because electrophysiology is moving into the age of light, where people are using microscopes much more. We're able to control electrical activity with light. And if you couple that with interesting new behavioral techniques, including aspects of virtual reality, you can start to do the most amazingly imaginative experiments, which I think will, in the next few years, really shed light on how actual neural circuits process information in learning and memory, but also in lots of other processes like perception and motor action and so on. So it's a great time for young people to be involved in the subject.
So I think it's really interesting to see that LTP has been brought into the modern age by combining physiological experiments with optogenetics and multiphoton microscopy. This is really exciting stuff. Fantastic.
What does it mean for you to be a Brain Prize winner?
It's an extraordinary honour, of course. I mean, one's absolutely delighted to be a Brain Prize winner, and particularly to share it with two old friends who've contributed so much to the same field.
It was a great surprise, first of all, but it's wonderful. And I'd like to think it's not just a prize to me, but to everybody I've had the privilege to work with.
I think it's also good because those of us who work on long-term potentiation feel that, yes, the rest of the community believe that this is an important process, as of course we have for our whole careers. Otherwise we'll be working on it. So it's good for that, and lots of reasons.
We were last night having a few drinks.
I can't remember. Were we?
You can't remember?
No. Well, as you know, alcohol interferes with the NMDA receptor and impairs the process of long-term potentiation. And I'm afraid that's probably what's happened to my memory, because I can't remember what happened.
You can't remember anything from last night? Okay.
Not very much, no.
I think we have some footage of it.
Oh dear, oh dear. Oh, that's very sad.
Obviously not very good. Obviously that Danish wasn't very good. Let's try again.